everyone. So we have discussed about the T cell activation. When any site infection is there, then if it is in blood, it will be drained into the spleen, and if it is into the tissue, then it will drain through the lymphatics into the regional lymph node, where they will be they will present this antigen processed by antigen presenting cell to the T cell in the paracortex region, and that will again this leads to the activation of the T cell, which will again secrete the cytokine that cause the activation of the B cell, which helps in the formation of the plasma, which converts that B cell into the plasma cell, and that plasma cell secretes the antibody into the circulation, and in this way the antibody formation takes place, and at the site of the infection the antibody goes and then try to clear that infection. Okay, so we have known about the activation of this T cell. We have discussed about the activation of the T cell. Any MSC class 2 protein or a class 1 that will comes with the antigen, then that will be checked by the TCR, then CD4 or CD8 depending upon the MSC class 1 or 2, that will be checked. Then they will be the this is the primary signal, first signal, then there will be a co-stimulatory signal, that is the B7 and CD28, and by which the T cell will be activated. The CD3 will send the intracellular signal and then release the cytokine that will release interleukin 2 that will act on the autocrine fashion and cause the expansion or proliferation of the T cell as well. So we have discussed about this first signal, then co-stimulatory signal, which leads to the activation of the T cell. Now let's discuss about what will be the fate of the T cell. Once the T cell is activated, what is going to happen? The T cell will be converted into the Th1 arm, Th2 arm or Th17 subset. The Th1 is due to the intracellular pathogen that is called cell mediated immunity. Th2 is actually a humoral immunity, or say antibody dependent, where antibody will formation. This will be because of the extracellular bacteria and pathogens, normally bacteria, parasites and fungi. So talking about the Th1, it will depends upon the intracellular organism, that will be the viruses, intracellular bacteria and other intracellular component, whereas the Th2 arm will be mainly responsible for the extracellular bacteria. Virus will be rarely found extracellularly, sometimes while passing through the fluid before reaching the cell, like if you have a hepatitis V virus, they will before reaching the hepatocytes, they will be encountered in the extracellular fluid. But you have to remember that this all intracellular will be processed by Th1 arm, that is cell mediated immunity. Then there will be a Th2 arm which will process the humoral immunity, or say antibody dependent. And there is a Th17 subset that has two functions. One, it secretes the regulatory protein, regulatory cell that will regulate the all this T cell functions, as well as there are some extracellular fungi and bacteria that will be controlled by chemotaxis of the neutrophil and phagocytic cell. Okay, so let's move through it. So talking about the development of the Th1, Th2 and Th17 subset, the helper T lymphocytes serve as the orchestra of virtually all the possible effector mechanism that will arise to destroy the pathogen. So the helper T cell is the one of the manager, one of the core component that will handle every section while dealing with the pathogen killing. The effector mechanism that are controlled by the TS cells include the antibody synthesis, macrophage activation and cytotoxic T cell killing. So there are three things. TS cell will lead to the antibody synthesis by Th2 arm. There will be the macrophage activation, that will be Th1 arm, and cytotoxic T cell killing, that is also due to the Th1 arm. Okay, that is cell mediated immunity. And if through the humoral immunity, or say there will be the release of antibody synthesis, and this all will be regulated by the that will be the controlled by the TS cell. Okay, the decision as to which of the following mechanisms should be engaged is based on the character of the invading pathogen. So what will be come out? It will leads to Th1 arm or Th2 arm or Th17. What will be the come out of from the Th0 arm? So the naive T lymphocyte, you say, what will be the fate of it? It will be Th1, Th2 or Th17. It depends upon the pathogen that is present intracellularly or extracellularly. The nature of the pathogens that will leads to the formation or proliferation or say differentiation of this Th0 into Th1 or Th2 or Th17. So this is entirely depend upon the invading pathogen, the characteristics of the invading pathogen, and is controlled by the differentiation of the specialized class of the helper T cell. So it is depending upon the invading pathogen characteristics, and that will control by this helper T cell. All CD4 T cell require recognition of their specific antigen complex to MSC class 2 by their TCR first signal and then co-stimulatory signal through binding the B7 on the professional APC. So we know the activation of the T cell. The activation of T cell will depend upon the antigen brought by the MSC class 2 or class 1, and that will check by this TCR as a first signal, and then there will be the co-stimulatory signal through which the binding of the B7 and B7 on the professional APC and CD20 on as CD28 as a co-stimulatory signal. So there are the two signals required. With one signal, or say firstly bringing of the antigen processed by the antigen presenting cell will check by the TCR, and then there will be a B7 and CD28 this combination by the co-stimulatory signal. So after two signal, this is get activated, and depending upon the nature of the invading characteristic pathogen, that will be differentiated. The three major classes of helper T cell that arise from the same precursor naive T lymphocyte is TS1, TS2 and TS17. The pattern of differentiation is determined by the antigen or the pathogen causing the infection. Again, the pattern is determined by which? By the antigen or the type of pathogen causing the infection, the cytokine produced in response to the antigen, and the transcription factors stimulated by the cytokines. So you have to understood the naive T cell, the TS0, will convert it into TS1, TS2 or TS17, depends totally on the type of the pathogen, the cytokines they produce, and also the transcription factor is stimulated by the cytokines. So we can see this TS0 will convert it into TS1, that is intracellular infection if there is that release the interleukin 12, that will release the TS1 arm, and that is the cell mediated immunity. In the cell mediated immunity, the cell should be killed, so that amplify this will reach in that release the interferon gamma. This will amplify the TH1 response, inhibit the antibody formation, TS2 response, activate the classical macrophage, and drives isotype switching to class G. So what is the main function of your intracellular infection is how we can deal with how this T cell will deal. It will activate the T cell mediated immunity, and after activating the cell mediated immunity, what will happen? This cell mediated immunity will release interferon gamma, and that will again amplify this in cell mediated immunity, that will inhibit other arm, that is the antibody dependent arm, and then activate the classical macrophage. So the initial macrophage become more and more active, and now they can kill. They can drive the isotype switching to immunoglobulin G. They activate the CD8 cell, okay, and by the which we can kill the intracellular pathogen by activating amplifying the TH1 response. If it is due to the infection extracellularly, that is parasitic infection, bacterial infection, then they will release this, activates the TH2 arm. This TH2 arm is release this interleukin interleukin 4, 5, 10, 13 and TGF beta. Interleukin 4 amplify this TH2 response. Interleukin 4 and 10, this will inhibit the TH1 response. So you have to understand what one thing is that they will release some of the cytokines that will activate their own arm and inhibit the other arm. So if you talk about the interferon gamma, this will activating the TH1 arm but inhibiting the TH2 arm. If you are talking about the interleukin 4 and 10, then this was actually amplifying the TH2 arm but inhibiting the TH1 arm. So you have to understand, once one arm is get activated, that will release the cytokine that will shut down the next arm, or say suppress the next arm. So because the one arm need to be more powerful. If it is TH2 response has been activated, that is this antibody dependent pathway has been activated, that will inhibit the TH1 arm. That we have to understand. And what are the responsible cytokines? That becomes the important. And the cytokine that amplifies the TH1 is interferon gamma, and that will inhibit this TH2 arm. The TH2 arm will be activated by interleukin 4, interleukin 10, that will actually amplify the TH2 response but inhibit the TH1. Interleukin 5 derive the eosinophil maturation, activates the alternate macrophage. Interleukin 4 and 13 derive isotype switching to hemoglobin E and alternative macrophage activation. And interleukin 5 and TGF beta drive the isotype switching to hemoglobin A. So these all are the function of your TH2 arm. If it is leading to the TH17 arm, that will be the interleukin 23, 6 and TGF beta. This will activate, what will do? This 17 activates the tissue cell and leukocyte to secrete the inflammatory cytokines, leading to the requirement of the neutrophil. So if there is extracellular bacteria, neutrophils should be come out and to the infectious site, which was due to the chemotaxis, and this after activation, this will again enhance that neutrophil going to the infectious site. Interleukin 17 and 22 acts on the epithelial cell to secrete the antimicrobials and improve the bacterial function. So these are the function of your subsets that will develop due to the type of infection. If it is intracellular, this is Th1 arm. It is extracellular, Th2 arm. One is activated, another is inhibited, another is activated. This will be inhibited, and that the cytokines will play that role. Okay. So this is the main function, or say main fate of your Th0, that is naive T cell that will come into the Th1 arm or Th2 arm or in the Th17. This also regulates this mucosal cells at the site of infection, and Tregs also secretes TGF beta, causing isotype switching to immunoglobulin A. This Tregs helps in the sometimes this is the controlling the inflammation, so shutdowning the activation. That is also can be 17 as a regulatory cell. You can think as the T cell, which is a regulatory cell. Okay. Now, differentiation of Th0 into Th1 arm cell is stimulated by the intracellular pathogen, that is a virus and intracellular bacteria. We have discussed about this. This pathogen including strong innate immune response with a result in the production of interleukin 12 by the macrophage and interferon gamma by NK cell, increasing the expression of the transcription factor for Tbet. In turn, Th cell secretes high level of inflammatory cytokines, that is interferon gamma, and does amplify the Th1 response, inhibit the Th2 response, activate the classical macrophage, and enhance the isotype switching to immunoglobulin G. That is the relation of your this is the function of your Th1 arm. The differentiation of Th0, that is naive T cell, in Th2 seems to be encountered in response to the extracellular parasites such as helminthes and allergen. Due to inability to phagocyte this pathogen, there is no significant macrophage or NK cell stimulation. In this way, naive T cell seems to produce interleukin 4 constitutively, and in the absence of interleukin 12 stimulation, so there is no interleukin 12 because there is no intracellular signal, so there is only interleukin 4. If there is interleukin 12, that is due to intracellular signal, that will goes to the Th1 arm. But if it is not interleukin 12 but interleukin 14, that will activate your Th2 arm. This will regulate the production of interleukin 4, encourage the differentiation of into Th2 by the induction of transcription factor, that is GATA3, and additional interleukin 4 will produced by the activation of the mast cell and eosinophil by the helminthes or allergen for the dividing the differentiation into the Th2 arm. The function, several cytokines are produced by the Th2, including interleukin 4, 5, 10, 13 and TGF beta. The interleukin 4 causes the lymphocyte to isotype switch predominantly to immunoglobulin G, which will bind to the mast cell, eosinophil and basophil. In collaboration with the interleukin 13 and 4, enhance the alternate macrophage activation for tissue repair and increase the intestinal mucosal secretion and peristalsis. The combination of interleukin 4, 10 and 13 together promotes the Th response while inhibiting the Th1 response, and interleukin 5 derives the maturation and activation of the eosinophil. The differentiation of Th0 into Th17 occurs in the presence of extracellular bacteria and fungal infection. Local cells reacts by infection secreting the interleukin 1, 6, 23, TGF beta, inducing the expression of the transcription factor, that is the ROX-ROY-T in Th17 cell. This activated T cell within in turn secretes cytokines like interleukin 17 and 22. 17 increase the chemokine production, recruiting the neutrophil. Interleukin 12 stabilize the interaction between cell endothelium, decreasing the permeability, and interleukin 17 and 22 introduce the secretion of antimicrobial by the endothelium. So we have discussed about that. There is another population of T cell that arises from the Th0 into T regulatory cell, that is Treg cell. T regulatory inhibit the Th1 cell function. So the main function of your Treg cell is to inhibit the Th1 cell function. I defined by their constitutive expression of the CD24, 25 on the surface and expression of the transcription factor, that is FOXP3. Secret inflammation inhibition cytokines such as interleukin 10 and TGF beta have been shown to be critical for the prevention of autoimmunity. So this Treg cell is responsible to prevent us from developing autoimmunity. If you have absence of this T-regulate cell, or say there is any problem in this FOXP3 or CD25, then we will develop autoimmune diseases. That you have to understand. This will also secrete inflammatory inhibiting cytokines such as interleukin 10 and TGF beta. So the main function of T-reg cell is to control the over activation of your immune system, or say T cell, and preventing the autoimmunity. Okay. Now, talking about the development of cytotoxic T lymphocyte, we know CD cell, CD4 or CD8 require both a primary and co-stimulatory signal to be activated. So, this T cells require two signals to be activated, which we have clear. The main difference between them is CD8 recognize specific antigen present by the MSC class 1, that is rule of 8, and rely upon the cytokine produced by the helper T cell to proliferate and ultimately differentiate into the cytotoxic T cell. So, how the cytotoxic T cell is formed? Cytotoxic T cell is formed mainly by the antigen that is processed and presenting by the MSC class 1 first, and if it is processed by MSC class 1, then CD8 cell will go, and after CD8 cell will go and see that it has been processed properly, then it will get activated, and this activation will be aided or facilitated by the helper T cell, and then this will convert it into the cytotoxic T cell that will kill the intracellular fungus, intracellular mainly viruses and bacteria, mainly intracellular pathogen, and this CD4 cytotoxic cell eliminate that intracellular pathogens. Now, talking about the clinical correlation about this TH1 and TH2 arm, if you know about the leprosy, that is the oldest disease, Hansen's disease, the tuberculosis versus lepromatous leprosis, there are the initial form and the advanced stage. The initial is tuberculous, the advanced is lepromatous is leprosy. The progression of disease with Mycobacterium leprae in human has a well documented example of the crucial balance between TH1 and TH2 arm. So, one is cell mediated, another is antibody dependent. Leprosy is not a single clinical entity, but rather a spectrum of disease with tuberculous and lepromatous form at the far poles. So, at the two different, one is initial stage, one is the advanced stage. In tuberculous, that is the initial stage, the patient has a strong TH1 arm. Initially, you got infected with this Mycobacterium leprae. This is the intracellular pathogen. Now, that's cell mediated immunity or TH1 arm will be activated, which eradicates the intracellular pathogen by granuloma formation. There is some damage to the skin and peripheral nerve, but the disease progresses slowly. If at all, the patient survives. So, the intracellular in the initial stage, and your own body will activate, that will kill your intracellular pathogen by forming the granuloma and limited the infection, and you will be survived. But if you have in the lepromatous stage, what will happen? This TH2 response is turned on, and the problem is when one arm is turned on, then another arm is inhibited, and because of that, now the disease is severe, because the killing machine, the killing arm, that is the TH1 arm, is not present in lepromatous leprosy, because now the TH2 has been kicked on. So, there is turn on. So, lepromatous leprosy, you can see this TH2 has been turned on. This, since you know from the previous knowledge which we have discussed recently, that if TH2 is turned, this TH1 will be inhibited. If TH1 is on, TH2 will be inhibited. Since now TH2 response is turned on, because of local reciprocal inhibition, the cell mediated response is depressed. So, TH1 is inhibited, and patient develops antibody to the pathogen that are not protective, and the mycobacteria multiply inside the macrophage, sometimes reaching the level of 1010 per gram of the tissue. Hyperhemoglobinemia may occur, and this case frequently progresses to disseminated and disfiguring infection. So, we are understanding